Hello everyone, and welcome to episode 52. So far in this series on the physiology of plants, I've discussed how plants develop and grow, how they extract water from the soil, and how they absorb and process nutrients. Today's episode will be about another fundamental quality of the plant's existence: their senses. Plants don't have eyes or ears like animals, but they do have a number of sophisticated internal chemical signaling systems and defensive responses. Plants don't have brains like us, or sentience, but they do have a unique kind of plant awareness about the world around them. They can detect gravity, so they know which way to grow their roots and their shoots. They can detect sunlight, so they know where to best position their leaves and allocate resources for growth. They also have to have some kind of awareness of the time of day, for example, so they know when to bloom, and an awareness of the time of year, so that they know when to drop their leaves and go dormant for the winter, or when to pump up investment in growth for an upcoming growing season. While this plant awareness isn't sentience or sapience as we understand it, plants are able to learn about their environment and make choices so as to maximally benefit its growth and survival in its environment. If this still seems kind of like a rather vague and unclear description, just get comfortable and relax, because I'm going to explore pretty much all of this stuff in detail. To describe these plant sensory systems as simply as possible, they have various types of specialized sensory cells, which detect some kind of input. The response from this input sends an intracellular signal to other cells elsewhere in the plant's body, and those cells on the receiving end of the signal will make the appropriate response. If they get a signal that temperatures are dropping, they might respond by preparing for winter. If they get a signal that gravity is this way, the shoot will grow the other way. I'll start by talking about this sense of gravity, because I find it particularly interesting. The ability for plants to detect gravity is called the gravitropic response. Do you recall the structure on the end of the root called the root cap? I briefly talked about this in the last few episodes, but to summarize, it's basically a shield of cells on the apical side of the root meristem. It acts as like a shield because it protects the fragile stem cells in the meristem. The cap is designed to be sloughed off and rubbed off against the rocks and the dirt that the root bumps into as it's growing, and the whole time it protects the meristem. The root cap has another function, besides just acting as a raw armor. The root cap possesses a group of cells in its center called amyloplasts. These amyloplast cells are partially filled with a handful of starch granules, like just little clumps or balls of starch. These are dense little blobs, and so they sink to the bottom of the cell's cytoplasm. Gravity will pull them down against some spot on the inside of the membrane of the amyloplast. Sensory proteins in the membrane are like pressure sensors, and they're used to sense where the granules are, and then inform the amyloplast, and thus the plant, which direction gravity is. And this passing along of information encourages the root to grow downwards. But how does it encourage the root to grow downwards? How does moving this information work on a chemical level? The signaling chemical in this circumstance is called Abscisic Acid, or Oxen, which is a powerful hormone that exists at extremely low concentrations, like a few billionths of a gram of Oxen per gram of plant tissue. When the root is growing straight down, Oxen is being actively transported throughout the plant's root tissue, down to the root through the center, to the tip, and then out to the sides and back up. This is a stable kind of Oxen flow that has the Oxen distributed equally by proteins throughout the root's tissue. However, when the root isn't growing vertically, the amyloplasts can detect this, and they tell the cells to rearrange their membrane proteins so as to move more Oxen to the downward side. Say the root is going along horizontally. Oxen will accumulate in the lower half of the root, like water in a cylinder lying on the floor, and this increased concentration of Oxen in the lower half will inhibit cell growth in the roots, on the underside of the root. Now this is kind of interesting, because Oxen is revealed to be a growth regulator. In steady, stable conditions, when all the cells get a steady amount of Oxen, they all grow at a steady rate, straight down. But when some cells get just dumped on with Oxen, their growth is inhibited. Conversely, the cells on the top half of the root are exposed to less Oxen. The concentration is reduced even farther, and their growth is relatively uninhibited. And so this creates an asymmetric growth, where the top side of the root grows faster than the bottom side. The cells grow larger, and this causes the root to curl and point downwards, re-establishing the vertical growth pattern. While gravity is a constant variable, unmoving and perpetually unchanging, other variables like light and temperature are very variable. They change throughout the day and from season to season. Plants have a variety of responses to a variety of different wavelengths of light. One of these responses is called phototropism, which is a basic response where the plant's body turns or bends to face a light source. Specifically, plants only engage in phototropism when they're being stimulated by high-energy blue light. The blue light excites electrons in a variety of photoreceptor pigments, which all induce different responses. Some of these blue light photoreceptors regulate flower growth and the opening of stomata. Specific photoreceptors, called phototropins, are pigments in the tip of the growing shoot structure that will absorb blue light. And by absorbing this blue light and getting an electron all excited into a higher energy state, the phototropin protein undergoes a conformational change. It changes shape, and this opens it up to phosphorylation. When ATP is used to phosphorylate the phototropin, the protein undergoes another conformational change that sends a signal to auxin transporting proteins, telling them to alter their distribution of auxin. In case you were wondering, yes, this is the same chemical auxin that helps roots bend downwards, but in shoots, auxin has the entirely opposite effect. In the roots, auxin is pooled on the lower side of the root, and this high concentration of auxin inhibits the growth of cells on the lower side of the root. Meanwhile, the cells on the opposite side, on the top, have their growth unhindered because of a relatively lower auxin concentration, and they grow larger. The combined effect will bend the root towards gravity. But in shoots, the phototropins send signals to the auxin transporting proteins to have them shuttle auxin only down the dark side of the shoot, down the side of the shoot that's not exposed to the blue light source. Because auxin here has the opposite effect as it does in the roots, this increased concentration of auxin in the cells of the dark side of the shoot will stimulate them to grow larger, and the low concentration of auxin on the bright side of the shoot will cause them to grow smaller and be inhibited. Just like this asymmetrical growth bends the root, this will also bend the shoot and causes the plant stem to bend towards the light. But these cells in the stem that are bending, they're already mature cells. They have cell walls. So how can they bend like this? The answer is proton pumps. But buckle up, because this explanation is like a water slide of chemical reactions that just keep coming, one after another, to orchestrate the plant's responses. Alright, so the signal sent out by the phototropins ends up stimulating the proton pumps, who begin fervently pumping hydrogen ions out into the cell wall itself. They go into the pores and the little open spaces between the polymers in the actual structure of the cell wall. The result of all this hydrogen pumping is a high concentration of hydrogen ions in the cell wall, and this makes the wall particularly acidic. The low pH will activate enzymes called expansins, and these expansins will go on to break a few of the hydrogen bonds holding the polymers in the cell wall together. This controlled degradation of the cell wall allows it to loosen up and become a bit more flexible. Now this is really important because the cell wall can't bend, but it can loosen to accommodate a membrane turgid with water. Consider the fact that the high proton concentration in the cell wall also makes the outside of the cell very positively charged, as all of these protons out there have positive charges. And in contrast, this will make the inside of the cell relatively negatively charged. This internal electronegativity ends up attracting positively charged ions to enter the cell, and this raises the cell's solute concentration. As I discussed in great detail in episode 50 on plant hydration, a high solute concentration creates an osmotic gradient that draws water into the cell. This incoming water will fill the membrane until it's taut and turgid and pressing up against the inside surfaces of its cell wall. But now add the fact that auxin stimulation made the expansins loosen up a portion of the cell wall. Now when the membrane reaches turgidity, the internal pressure pushes out against the loosened part of the wall, and the cell as a whole expands in size. Thus, auxin activates the growth of these cells on the dark side of the shoot, and causes the shoot to bend towards the blue light. Photosynthesis in plants is stimulated by wavelengths of blue light and red light, and red light causes its own set of responses. One of the most interesting responses to red light in plants is called the red, far red switch. One of the wavelengths of light typically found in direct sunlight is a red wavelength around 660 nanometers long. Far red light includes wavelengths closer to 735 nanometers, and this is the light that rarely gets absorbed. Because this far red light isn't readily absorbed by leaves, it's one of the only wavelengths of light that will pass through a lot of the leaves and make it to the ground. Thus, if a leaf is getting nothing but this far red light, if that's all the light that it can access, that's all the light that's hitting it, that will tell the leaf that it's in the shade, and that it's being blocked out by other leaves, perhaps leaves on other plants. And all of these leaves and all of these plants are all competing for sunlight. Where red light stimulates growth, far red light can suppress growth. The idea of a red/far red switch was created as researchers were studying the enzymes and the pigments involved in absorbing these wavelengths of red and far red light. They called these photoreceptor pigments phytochromes, which actually absorb both red and far red light, but only when they're in particular conformations. When the phytochrome pigment is in the P sub R conformation, it will absorb red 660 nanometer light, and this will change the protein's shape into the P sub FR conformation. When the pigment is in the P sub FR conformation, it will absorb far red 735 nanometer light, and this will change it back into the P sub R conformation. In this way, exposure to red or far red light is like a switch that moves these phytochrome proteins between conformations, between P sub R and P sub FR. These subnotations just reference the kind of light that it will absorb. The P sub R conformation will absorb red light, R for red. The P sub FR conformation absorbs far red light, FR. So what does this switch do? What does it turn on and off inside of the plant's body? When the phytochromes are in the R conformation, in the P sub R conformation, this means that they've been exposed to 735 nanometer light. They've been flipped into the R conformation, and the thing that flips that switch is exposure to 735 nanometer light. This exposure will send signals to the plant telling it that these leaves with the R conformation proteins are in the shade, that they aren't getting adequate light. As a response, some plants will simply stop giving sugar to the shade-covered leaves and let them wither and die, while investing that sugar in better positioned branches and leaves that have better access to light. Other plants will take this shade signal and interpret it to mean that they're surrounded by other plants or natural light-blocking obstacles, and so they should grow taller to reach out above them, to outcompete everything around them for the sunlight. The stem will grow and rise upwards, hopefully bringing the shade-covered leaves out of the dark and up into the light. Now on the other hand, when the phytochromes are in the FR conformation, in the P sub FR, this means that they've been exposed to red 660 nanometer light, and that sub-R switch was flipped to the sub-FR switch, and so they're now in the FR conformation. The phytochromes in this FR conformation encourage the plant to germinate and to flower. When a seasonal plant gets signals of fewer hours of more and more red-tinged wavelengths of light, caused by shorter days with a sun that rises lower in the sky, this tells the plant that it's approaching the end of the summer, the end of a growing season, which goes on to stimulate the processes that encourage sugar storage and seasonal dormancy. It takes a little bit more than some red light just to make a plant flower. Flowering is a really important part of the plant's life. It's involved in reproduction and the strategic timing of seed dispersal. It's important that flowering be timed with the day and with the seasons so as to maximize the plant's chances at reproduction. Thus, the signal to start flowering is more complex than simply being exposed to a particular wavelength of light. The length of time that they're exposed to light is also important. Plants that live in the tropics don't tend to see seasonal changes in light. They get about the same 12 hours of light every day throughout the year. But closer to the poles, you'll start to experience seasons. The winters are periods with less light and fewer total hours with the sun above the horizon, while the summers are the total opposite. you have more light with the sun above the horizon for more hours in a day. The amount of light that a plant gets throughout the year depends on where it's located on the planet, and this amount of light is called the photoperiod. Plants will respond to various changes in their photoperiod that are appropriate for the season. For example, a plant that experiences an increase in the photoperiod, which is an increase in the amount of time exposed to sunlight, will sense that winter is ending and summer is approaching, as the days are getting longer and there's more sunlight. Conversely, a decrease in the photoperiod means that the days are getting shorter, and this indicates that summer is ending and winter is coming. So there are some plants called short-day plants. These bloom during the times in the growing season when the days are relatively short, like the early spring or the mid to late fall. These plants are used to short days and long nights, and when these conditions are met, they'll flower. Other plants are long-day plants, and these thrive and bloom in the middle of the summer when the nights are short and the days are long. If you were to interrupt a long night with a brief but sustained flash of red light, say for about 60 seconds, you would flip the phytochrome protein switch and tell the plants that this time of year has short nights. You would make them think that their night ended when you gave them that flash of red light, and they would think that that flash of red light, that 60 seconds, they would think that that was a daytime period. As far as the plant can tell, the long night just got split up into 2 short nights by a little 60-second long day of red light. This false signal will confuse the plants by making them think that it's a time of year with long days and short nights. In these conditions, long-day plants will flower, and the short-day plants won't. Now let's say that a few minutes after flashing the plants with red light, you decide to flash them with far red light. Well, this will flip the switch back to the way it was originally, and the plants will bloom normally. You can keep doing this, showing the plants multiple flashes of red and far right. You can keep doing this, showing the plants multiple flashes of red and far red light back and forth throughout the course of a night, and wherever you leave the chemical switch, that will determine how the plants respond by either flowering or not flowering. But activation of the phytochrome isn't the only thing involved in flowering, as I was saying earlier. Plants have a circadian clock of sorts that involves cyclical increases and decreases in the concentration of various proteins and hormones as the day goes on. One of these proteins increases in concentration during the daytime, which stimulates a gene called constans, or CO, and this makes it produce an enzyme. This enzyme stimulates the expression of flowering hormones, but only when present in certain quantities. As the enzyme decays relatively quickly, its concentration in the cell doesn't rise very high. But when the phytochrome are activated by red light, they will stabilize this CO enzyme and prevent it from decaying. So it's still getting produced, but it isn't breaking down, so it accumulates, and the concentration of the enzyme in the plant cells rises. In the course of a long day, a plant will thus accumulate a lot of CO enzyme in its tissues. In a long day plant, this will eventually reach a threshold where the CO concentration begins to encourage flowering hormones like florigen to initiate blooming. But in short day plants, this high CO concentration inhibits flowering hormones and prevents blooming. The short day plants are sensitive to shorter photoperiods, and they only bloom when the time is right. As the plant experiences days and nights, its photoperiod stretches and shrinks as the seasons perpetually cycle through time. The plant ages. Different hormones are produced and expressed at different times in the plant's life. The first bit of growth that a plant sees is from its apical meristem, where the shoot grows up out of the ground. This apical shoot has what's called apical dominance. This means that the lateral buds that can form branches will actually be suppressed by the dominance of the apical meristem growing upwards. But when this dominance effect fades as the plant ages, or if it's shut down by some herbivore coming and eating the top of the plant, biting the meristem right off of it, the apical dominance is lost, and the lateral buds begin to grow outwards into branches, all with their own meristems at their tips. This dominance effect is created by the apical meristem producing Oxen, which is shuttled throughout the rest of the plant's tissue. I know that I've mentioned the chemical Oxen a lot in this episode. It helps roots find their way down, It helps shoots point towards the light, it helps regulate growth, and it sustains apical dominance. Oxen is a really remarkable organic chemical because plants use it for a huge variety of purposes. It's believed that oxen's most fundamental role is in giving the plant a sense of self-awareness. I don't mean this in the way that we have self-awareness, but more like this. Because oxen is produced at the meristems, it exists in highest concentration at the tips of the stem and the tips of the branches. The oxen is carried down through the cells of the branches and the stem to the roots, but so much of it gets diverted and used for other purposes by various cells on the way down that by the time oxen gets to the tips of the roots, it exists in very low concentration in the plant's tissues. I mentioned earlier that oxen in the roots is measured in the billionths of a gram per gram of plant tissue. The relative concentration of oxen in a particular cell can help the plant recognize where in its body that cell is in relation to the rest of its body and all the other cells. It can say, all right, this cell has this particular concentration of oxen. It must be, in my upper roots. Or this cell has this particular concentration of oxen, so it must be somewhere kind of out on a branch, closer to a tip. Something like that. It helps the plant have a sense of body awareness. There's a group of plant hormones called cytokinins, and these cytokinins flow in the exact opposite direction to oxen. Cytokinins are mostly produced at the tips of the roots, and they get carried up the plant to the shoots. But they're also expressed, to some degree, in places where cellular division is active, like inside seeds or fruits, the buds and the zones of division behind the meristems. These cytokinins play a critical role for plants on a cellular level. They participate in the regulation of the cell cycle, which is the chemical process through which a cell divides and replicates itself in a stable, healthy way. Without these cytokinins regulating cellular replication, fruits and flowers can grow malformed, the seeds botched, and the meristem growth stalled. There are other groups of plant hormones, like racinosteroids, which help stimulate plant growth, and ethylene, which is associated with senescence. In the case of racinosteroids, these are like plant growth hormones. Through means and mechanisms that are not particularly well understood, the racinosteroids are able to alter gene expression to regulate the size of plant cells during periods of growth, and thus the size of the plant, or the size of some of its tissues. The racinosteroids stimulate stems to grow tall and branches to grow long. Ethylene is like the opposite of the racinosteroids, because instead of growth, ethylene promotes ripening, and ultimately the rotting and the death of the plant tissue. Ethylene is the hormone responsible for telling fruits to begin ripening, which causes all manner of changes in the fruit to make it softer and smell and taste and look more appetizing to animals. The purpose of fruit is to put the seeds inside of an edible container, so that animals like birds and primates will eat them. This way, birds and primates can act as shuttles for seeds, transporting them far away from their parent plant and helping the plant species spread out and reproduce. When you see a really green bunch of bananas, these aren't ripe. They have low levels of ethylene. But as they ripen, and as ethylene is released, they become yellower. Their husk gets softer, and the tissue becomes sweeter and it smells better as starches are transformed into sugars. I've mentioned dormancy a few times now, like how plants will lose their leaves in the fall and go dormant over the winter. And in the spring, dormancy will typically end, and the plants will leave dormancy and resume growth with the freshly watered soil and more sunlight. The plants use hormone signaling mechanisms to detect the oncoming seasonal change, and to make the appropriate changes to accommodate it. In the fall, a hormone called Abscisic Acid, or ABA, is released from the roots to inhibit growth. In the later summer, when the air gets cooler, rain is less common. The ground will get drier, and eventually it'll snow, and that snow will create a layer over the ground that prevents it from really receiving a whole lot of liquid water. The point is that over the winter, the ground gets really dry. At the beginning of this prolonged dryness, the roots will notice this, and they'll interpret it to mean that the long drought of the oncoming winter is arriving. This will stimulate the root to release abscisic acid, or ABA, which travels up to the leaves and tells them to close their stomata so as to conserve water. ABA is a dominant signal that overrides the signal from blue light to open the stomata. So even though it might be a bright kind of fall or early wintry day and the blue light is telling the plant, hey, you got sunlight, open your stomata, let's conduct photosynthesis, the ABA from the roots is saying, nope, emergency shutdown, we gotta close these, we gotta conserve water, winter is coming. In the seed, ABA interacts with another group of plant hormones called Gibberellins to influence the length of dormancy and the timing of germination. The Gibberellins have an internal biochemical role opposite of ABA. Where abscisic acid inhibits growth, Gibberellins promote growth. Many plant seeds have enough sensory processing capacity that they can tell when conditions are prime for germination or when conditions are a little more disadvantageous. They can tell if the soil around them is moist enough or warm enough. Signals that tell the seed that it's not a good time to germinate will encourage the expression of ABA. The ABA will bind with specific receptors on the membranes of the seed cells, and it'll initiate a signal transduction cascade within the cells that activates specific transcription factors that then promote the expression of specific genes. In the case of ABA, these genes that end up getting expressed will inhibit seed growth and prevent germination. But when conditions are more favorable, when the seed can tell the soil around it is fertile and the time of year to grow is optimal, these signals will encourage the production of a gibberellin called gibberellic acid, or GA. When GA is released into the cells of the seed, it can activate genes that express an enzyme called alpha amylase. ABA, in contrast, suppresses alpha amylase expression. This is important because alpha amylase is an enzyme that will break down starch and turn it into bioavailable sugar. When there's a high concentration of alpha amylase in the seed, it diffuses into the endosperm and begins to break down all of the starch that's stored there. This will release a lot of sugar into the cells, which wakes everything back up out of dormancy and initiates germination. The sugar is used to promote the plant's germination and its growth, like the protruding taproot searching for soil, and the cotyledons that will grow and unfurl into leaves. Inside the cell, the concentrations of ABA and GA are in perpetual flux. The concentration of these hormones in the plant cells is important, because the concentration of the hormone is directly associated with whatever the hormone does. In this case, the hormones initiate signal transduction cascades involving signaling proteins and enzymes that help read and express DNA. So as the winter ends and the spring appears, environmental conditions will cause less ABA to be expressed. The concentration of ABA in the tissue will slowly fall, and its capacity to suppress the expression of alpha amylase is reduced. Simultaneously, these conditions will cause more GA to be expressed, more gibberellic acid, and its concentration in the seed tissue will rise, and this will further stimulate the expression of alpha amylase. Gradually, the concentration of GA and alpha amylase will more and more overshadow the concentration of the ABA and its inhibitory effects, and sugar will begin to diffuse throughout the seed tissue as the alpha amylase breaks down all the starch. Thus, germination slowly ramps up as sugar accumulates and the cells in the seed reawaken with life and begin using all of that sugar to power their growth and their replication. One of the last things that I want to talk about today are the signals that plants use to detect and respond to infection, and also how they respond to the mechanical damage caused by an herbivore coming by and chewing them up. When a plant gets infected with a bacterial or a viral pathogen, its first line of defense is to close its stomata in the infected area to prevent further infection. This is the first step in the hypersensitive response, which is the plant's defense against these kinds of microbial invaders. After the stomata are closed, the plant produces toxins in the local area that will fight off the pathogen, much like the animal immune system. This even involves the chemical reinforcement of nearby cell walls, so as to create a literal cellular blockade to mechanically contain the pathogenic particles. Even more radically, the plant can sometimes engage in a scorched earth policy by killing its infected cells to deny their metabolic products to the pathogen. As this hypersensitive response is going on, the plant is also sending out warning signals to nearby cells, which initiates a slower but more widespread response called SAR, the Systemic Acquired Resistance. This primes all the other cells in the plant to release defensive chemicals at the first sign of pathogen infection. On the topic of herbivores, one of the most common but nutritionally and energetically expensive means that a plant has to repel a potential herbivore is by producing a chemical substance that tastes bad or smells bad, or that stings or is uncomfortable in some way to be around or to ingest. For example, the oils that are produced by herbs like lemon, sage, and peppermint will repel insects, which try to come and eat their leaves. The oils create intense odors and tastes that are so strong and so sharp that they fry the little insect's sensory buds and scare them off. Some pine trees will outwardly secrete a chemical called pinene, which poisons any bark beetles that might have been burrowing around in the tree bark. A lot of plants will express proteins called proteinase inhibitors. As the name suggests, these proteins disable the enzymes that break down proteins as part of animal digestion. As a result, if an herbivore eats too much of a plant, it will ingest too many of these proteinase inhibitors, which will lead to illness. The animal is thus discouraged from eating that plant, or at least from eating too much of it in one sitting. If the animal tastes a plant with a high concentration of these proteinase inhibitors, the animal will be disgusted by the taste. It'll be really turned off by it, and it won't want to eat the plant. What's crazy is that if plant tissue, like leaves, is getting physically destroyed by the mechanical chewing action of an herbivore, that pulped up, wounded tissue is able to release a protein hormone called cysteamine. Cysteamine is moved through the phloem, where it comes into contact with numerous plant cells and activates receptors on their cell membranes that initiates the synthesis of Osmotic acid. This Osmotic acid then goes on to stimulate the expression of various genes, including the gene that produces the proteinase inhibitors. So, by eating some of its leaves, an herbivore can cause the plant to release a chemical that will eventually make its leaves taste terrible. So, after just a few bites, the animal is encouraged to move on to a different plant. But this gets crazier, because some plants will even release an airborne signaling chemical that tells other plants nearby, "Hey, I'm being eaten over here!" And this causes those other plants to produce more proteinase inhibitors and defensive chemicals. Some plants will even release chemicals that attract predatory insects, like wasps, and these wasps will then come in and hunt down whatever herbivore is eating the plant tissue. Plants will often do this to repel caterpillars. They'll release these airborne chemicals, the wasps will be attracted to the chemicals, and they'll see these caterpillars eating the plants, and because they naturally prey on caterpillars, they are inclined to attack the caterpillar and kill it, or knock it off the plant, and this benefits the plant. It's a really kind of complex, intense kind of interspecies defense mechanism. Before I end the episode, I want to mention one more defensive measure that plants use. Sometimes, plants will produce a chemical that has a physiological and neurological effect on the animals that consume it. The idea here is that this effect is uncomfortable, and it discourages animals from eating those plants. But in some cases, this can backfire. Caffeine, cocaine, opium, and nicotine are all plant-based chemical compounds that are intended to scare the herbivore by disturbing its sobriety with a mind-altering chemical. The plant is saying, "Don't eat me, or I'll mess with your perception of reality." For most animals, being stoned or incapacitated can be dangerous, because they can't readily defend themselves or maintain sufficient awareness of their surroundings. They're vulnerable. There are some animals, however, that enjoy eating these plants, and they enjoy the psychotropic effects that the plants have on them. Humans, for example, will cultivate these plants and consume them, specifically to experience the mind-altering effects that they give. This includes everything from the coffee you drink in the morning to give you that kind of alertness and stimulation to get work done, to the joint that you smoke in the afternoon to relax, and the cigarette that you have on your work break to de-stress, to the opium products produced from Afghanistan's poppy fields, to the cocaine produced from the jungles of Colombia. All of these drugs are plant products that the plant evolved as a way of scaring off or repelling the herbivores who wanted to come and eat them. With that said, that's about all that I have for you today on plant signaling. I hope you enjoyed this episode, and I hope you learned something cool, something that you found interesting, something that makes you look at plants in a whole new way. If you liked this episode, give it a like and share it with your friends. If you like this series on plant physiology, then stay tuned for next week's episode on plant reproduction. And as always, thanks for listening.
Would you like to support the BioLogic podcast? It's super easy. When you open a new episode, press the like button or share it with your friends. If you aren't subscribed, you should hit the subscribe button so you can enjoy a new episode every week. You can also peruse our official store, which has a ton of cool stuff like hand-designed t-shirts, hoodies, and stickers. All the links you need are in the description section below.